I can really speak to this, Dr. Anderson. I really want to thank you for all the work you do on autism, and really, thank you for everything you're doing, and including this live presentation. So parents and other doctors and researchers around the world can really get a better understanding of your work. We're very appreciative of your work and for sharing your findings with us. So thank you for that.
Thank you.
Yeah, so Dr. Anderson has a very impressive background, which I'm going to try to summarize it before I pass the ball over to him and to begin with the presentation today. Dr. Anderson is a Harvard Medical School professor in the PhD program. He is the director of neuropathology at the Beth Deaconess Medical Center here in Boston, and he is also the lead neuropathologist investigating the cellular, molecular, and genetic mechanisms that could lead to the onset of autistic symptoms and behaviors of the what is called the Autism BrainNet, which is basically a bank of postmortem brain tissues of people with autism and also neurotypical people that have died by natural causes and whose brain have been donated for research. Dr. Anderson won the International Distinguished Dissertation Award as the top PhD thesis in science in both US and Canada, which is awarded every 5 years, so very, very impressive. And his postdoctoral fellowship at the MIT has led to findings in molecular genetics, brain slides, electrophysiology, synaptic physiology, in vivo electrophysiological approaches, and behavioral neurosciences. And this is very important because currently at Harvard, his laboratory leverages the technologies I just mentioned to investigate the molecular basis of genetic forms of neurological disorders, including epilepsy and autism spectrum disorder. So in today's presentation, Dr. Anderson will be sharing his, for us, a simplified version of his autism research and his recent findings analyzing brain tissues of people with autism, where potential biomarkers, specifically healthy brain tissues that are abnormally attacked by immune cells, could really provide insights into the origins of non-genetic forms of autism, which seems to account for a very large percentage of autism cases, especially those driven by what could be soon confirmed as a neuroinflammatory autoimmune disorder. So without a question, this is very groundbreaking research and one that could provide very, very critical insights into the origins and potential treatments for autism spectrum disorder. Last thing I want to mention before I pass the ball over to Dr. Anderson is we're going to have a Q&A session at the end of the presentation. We've actually received several questions already before we get started. So, and if anyone wants to ask any additional questions, so please provide those in the comment section of this video, and I'll do my best to summarize those questions and to fill them at the end of the presentation. So without further ado, Dr. Anderson, please take it away.
Great. Well, Enrico, thank you so much for this opportunity. And I really appreciate all the enthusiasm we've gotten around this new finding. So I'm going to share my screen and start the talk here. I'm going to touch on a couple perspectives. One is kind of the large chunk of my laboratory's work, which has been trying to bridge between some of the genes found in autism, the molecules, up to circuits, up to the behavior. And then just for perspective, and then I'm going to get into this new finding in the human postmortem brain and talk a little bit at the very end about how we're trying to bridge all those findings together. Let's see here. Okay, here we go. Okay, so everybody here is quite familiar with autism. It's really a behavioral disorder. It's defined by the behavioral symptoms. The incidence, this is even already out of date. It's even higher than this now, 1 in 53 and possibly more. It's more common in males than females, which is still somewhat of a mystery why that's true. And at least based upon, I would say, multiple pieces of evidence, the genetics argues for heterogeneity. The severity of symptoms and comorbidities varies from case to case. So we think it is a heterogeneous disorder. But there's surprisingly, at the very end, I'll show you some pathologies that might be present in a fairly large proportion of cases. Also, in the genetic heterogeneity, we think that there's pathways that are starting to come together. Ultimately, I'm hoping that the thing I'll show you at the end converges with these molecular pathways that I'll show you at the beginning. So the symptoms begin very early in childhood, and people are trying to push that boundary even further with new molecular markers and things so that you can really start treatment early. The core symptoms in the behavioral domain are reduced sociability and communication, increased repetitive behaviors, and restrictive interests. But really, there's also very important comorbidities that variably are present across cases, some irritability, tantrums, self-injurious and aggressive behavior. We're trying to understand the circuits underlying the social deficits and also some of this symptom as well. There's also variable amounts of epilepsy, maybe a third to 2/3 of the cases. There's interictal spikes that you can even see on the EEG in some patients that don't have a full-blown clinical presentation of epilepsy. And sometimes, unfortunately, this can lead to premature death related to the epilepsy. This is, in fact, what we see in some of the cases that have been collected in Autism BrainNet. There's also intellectual disabilities, various kinds, language problems, as well as other motor skills. So I guess what I sort of like to think about our work is kind of a disease mechanism sleuthing, as you know, traditionally in the realm of a detective. And I'll just kind of show you how we sort of think through this understanding of disease mechanism. So we started with the genetics in a way because genetics gives you a very solid foundation on which to build. And one of the earliest 1997 genetic things was the 15q region, which is duplicated, always inherited from the mother. So that was back in 1997. And then in more contemporary high throughput genetic methods, this is just an example in Nature in 2009, looking at copy number variation. So deletions or duplications of all the way across the chromosomes. They find relatively rare cases of deletions and duplications. And this same genetic change popped up again as the most frequent duplication in 2 different case series and absent from the controls in autism. And interestingly, the second most common CNV, Neurexin-1, which is a synapse protein, is deleted in a large number of cases, and GRID1 is also deleted. And the reason I show you these, highlight these 3 things is that I'll show you that they converge. And again, this is just a sort of in kind of the source of the behavioral deficits in autism before we get into this sort of immune mechanism, just because we have a long foundation in that direction. So here is how we have to think about autism. So we know that the brain is the source of behavioral phenotypes. You know, our normal ability in behavior and thinking and cognition, but also the deficits that arise in the behavioral domain come from the brain. And there's long range connections between these neurons. That's one of the unique features of that cell type. There are these microcircuits, and you can see there's incredibly large numbers of each of these little elements, which really contributes to the complexity of how we behave and how we think. So there's cortical columns, there's neurons, and then there's even this sort of structure, which they estimate, you know, 10 to the 10th of these little synaptic connections between the neurons, how they send signals between them. And this occurs through neurotransmitters and synaptic vesicles that are released. These bind to postsynaptic receptors, and then that excites the postsynaptic cell to carry the signal across these long-range axons to the next target. And what I'll show you is that we have convergence of multiple genetics into a transsynaptic complex involving Neurexin-1 on this presynaptic side, GRID1, which is a glutamate receptor delta subunit on the postsynaptic side, and then that forms synapses through this bridging protein called cerebellin-1 that binds this molecule and this molecule. And importantly, the gene that we're focusing on, UB3A, in that 15q region, is repressing the expression of cerebellin. So this is a really nice convergence of a pathway in the genetics of autism. These become useful small molecule or other approaches to treating the behavioral symptoms potentially in autism. But also, I feel that if there, and I'll show you pretty convincing evidence that there is an inflammatory component in a large proportion of the autism cases, we believe that this may converge through the same molecular pathway and are sort of actively looking into that. Interestingly, there's also convergence of the seizures that are frequent in autism, where that will also repress cerebellin expression to break this synapse. And it really depends on this gene UB3A. Again, not only sort of a, you could call seizures an environmental or, sort of acquired symptom that variably happens in case to case, converges in the same kind of molecular pathway. And I'm not going to get into it in great detail, but what's really nice about UB3A is that it's deleted and duplicated in two different forms of autism spectrum disorder, genetic forms. One of which is Angelman syndrome. Some people would put that into just a neurodevelopmental disorder with severe intellectual disability, severe language deficits, and not necessarily autism, because it has an unusual social feature, which is what looks like increased prosocial behavior. And that is exactly reciprocal to the reduced social behavior that you see in a large proportion of autism cases, and somewhat used to define the autism spectrum disorders. So we thought that this gene was really important because too little in Angelman syndrome, where it's deleted, always inherited from the mother, produced all these problems, but also increased social behavior. Whereas too much of this UB3A gene, which actually the most common form is a cytogenetic form here. This is cytogenetic spread, where we have 2 copies of every chromosome. Well, here's the 15q chromosomes, and you have this one little extra piece here that is an inverted duplication of itself in this small little segment you can see at the top there on the right. And the major gene we think is responsible is UB3A, because mutations in that gene cause Angelman syndrome, inherited from the mother. And this maternal inheritance happens for both conditions. And UB3A is the only gene in this region where in neurons, interestingly, the paternal copy has been completely shut off, so that only mom's copies are being expressed. So if it's mutated and inactive, you're going to get Angelman syndrome. If you get extra copies of the maternal inverted duplication piece like this, you're going to have like a tripling of the dose of UB3A. So that was kind of the foundation on which we built kind of a genetic line of research. And we were, interestingly, the first group to show that deficiencies of UB3A does cause hypersocial behavior in a mouse. So you can recreate many of these symptoms that had already been shown, but nobody had shown the enhanced social motivation or a desire to socialize. And it's interesting because people might wonder how useful is the mouse for such research, and it really allows you to do sort of detailed molecular and circuit-based studies, and really pilot out the mechanisms before you bring it to the clinic. And sure enough, yeah, their mice are hypersocial, a little bit like the social behavior that you get when a mouse is isolated. So when a mouse is deprived of social behavior opportunities, they become, I guess you would say the word lonely. They want social interactions, and they become very, very social with the first encounter with a new mouse. So single housing a mouse is a little bit like the effect of loss of UB3A. Whereas actually, I don't know about you, but after a very intensive social experience like a party or a concert or something, you often just want to sit by yourself and read a book. I think that the social behavior is an intrinsically plastic element of our behaviors, and here this gene is acting to repress it when there's too much of it, actually. I'm not going to show it to you, but we also have found some irritability and aggressive-like behavior in these mice as well. And we've almost finished the story around what circuitry UB3A is acting to promote that phenotype. And in fact, some of you may have children on this medication called risperidone, are the circuit where risperidone was effective in the mouse in tamping down the aggressive behaviors, but also altered the circuit that we discovered was key in a way that would repress these behaviors. So it really fits together with the medications that are currently used for that behavioral symptom. Really, this kind of molecular sleuthing, just to give you a flavor of it. I don't want to get into the details too deeply, but we took this gene now that we thought was really key because too much of it did produce the autism-like symptoms, too little produces Angelman's-like symptoms, including the hypersociability. So sociability was strongly toggled. I mentioned aggression was as well, but I'm just going to talk about the social circuit that we sort of broke apart a little bit here. So where in the cell is it acting? The cell has a nucleus, it has these synapses, it has all sorts of structures. Where is it acting? Well, I'm just going to give you the answer. By engineering this UB3A, we showed that it acts in the nucleus to repress social behavior. This led us to what happens in the nucleus. Well, genes are regulated in the nucleus. All the proteins that your body expresses come out of a coding sequences of the DNA in the nucleus. So we thought that, well, it must be a perturbation of the expression of those genes that is causing this impaired social behavior. That led us to looking at the genes, and I'm not going to get into the detail again, but we found that as UB3A goes up, genes are repressed. And if you take away UB3A, which is Angelman syndrome, genes go up. So here's the normal gene level. This one's repressed by too much UB3A, and it goes up when UB3A is taken away. So we saw 400 genes showing the same impact of being repressed by UB3A in this sort of reciprocal dose-dependent manner. We saw 200 genes that go up. Okay, so that's a total of 600 different genes. So how do you find the molecular pathway that's critical to autism? And I kind of gave you the sort of punchline at the beginning, but let me show you how we got there. So again, what is the specific gene target by this nuclear UB3A acting to impair sociability? And I'll also tell you this same pathway is leading to the aggression as well. So it's not just in social behavior, it's also in the aggression phenotype. So we actually took a technique which is very powerful, which is autism gene network analysis, where you can look at how proteins interact with each other. You can look at how they act through phosphorylation, dephosphorylation, these modifications of other proteins. But we just looked at how they physically interact with each other by binding. And we looked at the genes regulated by UB3A and this large collection of autism genes now that's growing on the Simons Foundation Autism Research Initiative website, Safari Gene, very useful tools. And you could look at these little blue lines connecting genes that are in blocks or circles, and the things that are downregulated are in green by UB3A, red is upregulated. And you can see that you get these little interacting clusters that are tightly interacting with each other, and there's a way to segregate out the strongly, tightly interacting clusters, and we got this set of 12 clusters. And you can see what their function is by looking at the set of genes. And again, there's really wonderful annotation of all the genes, what they might do. And there's some that are involved in gene regulation, some in glutamate synapses, so on and so forth. Well, we really focused in on the glutamate synapse gene set because, as I just showed you, the second most common CNV in autism is Neurexin-1, deletions of Neurexin-1. A very common one is also GRID1 deletions. And I just remember green is a gene that's repressed by UB3A. So this goes down genetically, this goes down genetically, and this is repressed by the excess of UB3A. So this, again, is a transsynaptic complex where UB3A is acting. And then, I'm not going to get into it, but some people had studied that complex a bit. And it's in very strategic parts of the brain. This is a mouse brain where the dark stuff is where it's located. and activity, increased neuronal activity. So when you have a seizure, your neurons are firing off a lot. And normally when we go out and learn something new, we hear a lecture, we talk to somebody we just met, some subset of neurons are active, right? Well, a seizure, too many of them are active, and essentially the whole brain has a traveling wave of neural activity that's discharging. Well, that activity, just like the increases of UB3A, repress cerebellum gene expression. And that really led us to thinking about this fact, which is that many genes that cause autism also cause epileptic encephalopathy. There is a very large set. If you go to the one website for early infantile epileptic encephalopathy genes, they also cause autism. That's arguing that the epilepsy may be, tightly linked to at least some cases of the symptoms that cause autism. And some of you may have at first-hand experience with this, is that if your child does have epilepsy and they're on medication, that their symptoms will sometimes improve when you get the seizures under control. I'm not going to talk about this in detail, but sure enough, the seizures interact with this whole molecular pathway involving UB3A and cerebellum, leading us to this model here. Okay, so really the next puzzle that we wanted to solve was which circuit in the brain. Remember all those levels of complexity, right? From the whole brain to these connecting elements, to all these little nuclei containing different neurons, to the synapse. Well, we talked about the cell, we talked about the synapse and the nucleus in the synapse. What about a circuit? Well, what's really convenient is the circuits are defined by the synapses, and there's various tools that you can do targeted deletions in particular cell types and targeted deletions and expressions of these genes in particular circuits. And we did that essentially. And that led us to figure out that essentially it's a defect in a reward pathway in autism. And this is our publication here. A lot of data there, probably too much for people to want to really pursue, but generally we've mapped it out to a ventral tegmental area, a mesolimbic reward circuit. So it's the same circuitry that leads you to all the rewarding things that you do, whether it's having a piece of cake at the end of your meal, or maybe you like jogging and you're sort of get pleasure out of jogging, or your social interactions, they're very pleasurable. We think that this is broken in autism, this sort of rewarding circuitry. And in fact, we found, a lot of people think of dopamine, right? And reward. And, cocaine, which is a drug of abuse, cranks up the dopamine level, so you feel very rewarded in an artificial way that sort of exceeds any kind of natural rewarding experience. Well, interestingly, there are also glutamatergic cells that form these glutamatergic synapses I was just talking about in that rewarding circuitry. And we discovered in this paper that if you toggle off those cells in this one little part of the brainstem, you'll shut down social behavior. If you crank them up, you'll make mice hypersocial, a little bit like the Angelman's mice. Or a mouse that's been deprived of social experience, they become very hypersocial. Well, this is where all this stuff is converging, where UB3A is repressing cerebellum and seizures are impinging on the brain to impair social behavior. And this is just an example of a tool that people now have where you can put in a receptor that binds to a specific drug, and you can make the neurons more active, you can inhibit the neurons in very particular parts of the brain. And this will, in the mouse, we demonstrated that this will crank up or down social behavior and rescue the social deficits produced by seizures or by excess UB3A. Interestingly, following our paper in 2017, another paper in 2018 published in Brain looked at the fibers that are connecting the midbrain. That's the area where I was just talking about, where there's dopamine and glutamate neurons. And those project forward into a number of different parts of the brain. It's called the mesolimbic reward pathway. Well, that pathway has fewer fibers coming out of it in autism in this study. Quite interesting, fits with sort of this genetic thing that led us to that molecular pathway. Okay, so now on to the new findings. So if you look at in this really nice review in Nature Medicine, somebody sort of synthesized all the genetic forms into one kind of graph here, and these are the exome mutations, just the most frequent genetic forms. These are the copy number variations where large segments of chromosome are deleted, duplicated. This is the one we're studying. This is the Neurexin one, so they're all in there. And then if you take some sort of syndromic disorders, tuberous sclerosis, Fragile X, and neurofibromatosis, fetal Lamb-McDermott, which may overlap with the SHANK3 pathway, these actually have autism symptoms. You know, again, it's behaviorally defined disorder. They collapse, only accounting for a very minor fraction of all of the autism that's out there. And some argue that maybe there's common inheritance, so there's some sort of genetic risk factor to developing autism. But again, that really requires a second hit mechanism to explain that. So what is explaining all this unaccounted for autism that is sort of, you know, maybe modulated by genetics, but not, at least the genetics that we've figured out quite yet. Well, this is why this program that I'm participating in, called Autism BrainNet, I think is very important. And I'll show you the really interesting findings that we made. And I made these findings serving as the clinical neuropathologist for Autism BrainNet. So if somebody passes away, the family, the physicians that are taking care of the individual, and sometimes they're trying to understand the cause of death for many, many reasons, even what they had during life. You know, we will look at the brain and try to understand that. And it's very commonly used in people with dementia, because we're trying to understand really what caused their dementia. Was it Parkinson's, Lewy body disease, Alzheimer's, or one of the many other causes like vascular pathology or something? So that really is what I do. I do for this whole network, all the cases that come in where somebody requests a neuropathology report, I provide that to the clinicians as well as to the family for the cases. And in that capacity, we found some very interesting adaptive immune system pathology that people had not really reported previously. And I'm just going to touch on this real briefly, which is that the immune system has two arms to it. There's an innate immune system, which is present in all living species, particularly, well, not just vertebrates, actually even before that, even, like a worm has an innate immune system. Whereas mammals develop this thing called the adaptive immune system. And so these are, the innate immune cells here. You can see macrophages, neutrophils or polys, and eosinophils. You may or may not have heard some of these cell types: mast cells, dendritic cells. And then there is this adaptive population, which are the B cells. They produce antibodies. And the T cells, which carry on their membrane a antibody-like molecule and these antibodies and T cell receptors, it's called a T cell receptor, is really the key trick that was evolved in the species to allow you to recognize hundreds and thousands of distinct molecules that are carried by viruses, bacteria, and other things that would otherwise hurt us, such like COVID-19 is one example. And this adaptive immune system, through somatic recombination, generates a incredibly diverse population of antibodies and T cell receptors. In fact, the person that trained me at MIT, Susumu Tonegawa, for my postdoc, won the Nobel Prize in this work back in 1987 for just demonstrating how that complexity was generated. And of course, these two systems communicate with each other. The innate immune system communicates with the adaptive immune system. And when there's an adaptive immune response, these cells participate in that process. So if this is happening, you're going to recruit macrophages. You're going to trigger all these cytokines and chemokines to go up. How is the diversity created? Well, in the genome, and this was kind of the award-winning, Nobel Prize award-winning work that he had done, is that there's this complex set of genes, exons, and they recombine. They can select many different options. So it's like a mix, like creating a dish with many different flavors in it. You can create this diverse population of receptors that ultimately come out. And there's a couple other tricks that they have as well to generate this incredible diversity. Now, this is very helpful in the case of pathogens, right? Viruses, bacteria, but sometimes it goes awry, and this immune system attacks your own cells. And remember, this immune system has to clear cancer as well. So that's another big job that it has. And in the case of cancer, those cells are coming from you, and some of the markers on those cells may actually be you. And when it's trying to clear the cancer cell, it picks up an autoimmune reaction, and that's often called a paraneoplastic. So in conjunction with a tumor, you may develop an autoimmune disorder. And we published on some examples of that in glioblastoma and various other things. So that's an important thing to think about as well. Okay, so here's the autism postmortem brain data. And this was back in 2005, coming out of Johns Hopkins, the Pardo laboratory. And for me, this was incredibly influential in changing how I think about autism. They found that cytokines, these innate immune reactive molecules, were dramatically elevated. And there is some adaptive immune related molecules that were also elevated, as you can see here. Chemokines were elevated, you can see many of them here, and growth factors and differentiation factors, all of which increase when there's an inflammatory reaction, sometimes to repair the injured tissue in response. Okay, so, and this was a sort of a random collection of cases of autism that had passed away and there was tissue available. So in order to see a signal like this, this must be present in a fairly significant proportion of those cases. Otherwise, you wouldn't have seen a signal. That was actually later confirmed in 2011 by the Geschwin lab at UCLA, using a different approach. Instead of looking at protein levels, they looked at transcripts, so the gene expression that generates these proteins. They found, again, an immune type response, mostly innate immune, inflammatory response. Interestingly, there was an antigen presenting and processing, which again is the innate immune cell getting ready to present an antigen to the adaptive cell. So they were enriched for elevations in those molecules, and that's in red here. And you can see that compared to the controls, only a subset of the cases, but a large proportion were elevated for these markers. And somewhat reciprocally, there was a decrease in neuronal markers, some of which are involved in these critical synapses that we were talking about before. Another group at Hopkins, Dan Arking's lab, found the same thing with microglial markers. So again, that's an innate immune system cell of the brain. Type 1 interferon signaling, here again that came up, the interferon gamma. That's kind of a viral immune response molecule, but also in a variety of other immune reactions, including some autoimmune. Is there other evidence for autoimmunity in autism? Well, there is a subset of cases. Before I get to that, this data shows that there's a family history of autoimmunity in a increased proportion of cases of autism. And in other autoimmune disorders, the MHC HLA molecules are altered, and there's an enrichment of specific MHC or HLA molecules in the individuals that have the disease, like multiple sclerosis. And the reason for that is the HLA is the molecule that presents this foreign antigen that the T cell or the antibody has to recognize. So particularly T cells. So that's a little bit of a hint. And then the autoantibody literature, you can see that, early on there thought to be a lot of them. And in fact, there still are in certain antibodies and other papers that actually aren't even listed here, like the folate receptor alpha. But here you can see very specific bands showing up with serum derived from the mom that recognizes fetal brain tissues run out for proteins, so that you can see what protein bands are stained. And some of these molecules they've sort of further defined. And so you get a whole cascade of antibodies, but this was only in a small subset of cases, maybe 7 to 10% of cases. But again, some of the other ones are more abundant. Okay, so this is the introduction to the data I'll show you that we've discovered in the postmortem brain. And I just want to remind you of what innate cellular immunity looks like. So what happens is there's many different subtypes of T cells. So there are the T cells which have the T cell receptors I mentioned that produce this repertoire, diversity of T cell receptors, so they can recognize this little thing called an epitope, which is a piece of protein, a peptide antigen that is in the virus, for example, that they need to get rid of. So it's a peptide that your body doesn't normally have. And it could be coming from a virus, it could be coming from a bacteria, a variety of things. The cells will process that into little pieces, and then on the HLA molecules, the MHC molecules, they'll present it on the surface, and then the T cell receptor can bind to that. And then that will draw in the cytotoxic T cell, for example, that has these cytotoxic agents, perforin, which pokes holes in the cell, or granzyme, which gets actually inside the cell and starts to break down the cell. And essentially, if you have a virally infected cell, in order to clear that virus, the best way to do that may simply be to get rid of that particular cell in your body. And again, COVID-19, that's actually probably happening. There's pretty good evidence for that in certain organs in your body. Well, here's what happens is that the cell dies often, and and this is really not an accurate cartoon of what the membrane looks like. This cell goes on to target another cell. Well, here is a image that I've found where a T cell, cytotoxic T cell, attacks a tumor cell. So this is cancer, and there's a recent Nobel Prize was the discovery that cancer cells are repressing the immune system. That's how they evade the immune system. And your body normally has mechanisms to evade the immune system. For example, during pregnancy, there are these same molecules, PD-1, PD-L1, expressed on the placenta because you want to keep the mom's immune system from going in and attacking the fetus as though it was a cancer or something. So here's a T cell that's having success at attacking the cancer cell. Somehow this was not, successfully inhibited. And what happens is you get this evagination of these little round bubbles off the surface of the tumor cell. And these have been called blebs. So they're called these membranous blebs, and you can see they're variable in size. Okay, so remember that morphology. Okay, so here is the surprising first finding, which is that in autism, I was starting to see, so this is a histologic section seen under a microscope, magnified, and you can see this is the white matter. So there's gray matter and white matter of the brain. This is the white matter where the myelin sheaths are formed. This is a blood vessel. This is the perivascular space between the brain and the blood vessel. Inside the lumen of the vessel are red blood cells. They're literally red in this stain. And you can see these little black dots in the wall of the blood vessel here. You can see a few here. This is a control. Here's another control where you can see a few black dots. These are the most abundant cases of lymphocytes. These little black dots are lymphocytes in the control cases. So we search for the most abundant lymphocyte blood vessels in all the sections from the control cases that we had created. And then in the autism cases, these are the ones that are most abundant in the autism cases. And you can see many, many more of these black dots. And the staining, and some of these are a bit older slides, and that's, that we had gotten from a brain bank. That's why the color is faded, but you can still see these same lymphocytes across all the cases. And interestingly, it was in a couple genetic forms of autism, P10 heterozygosity and XYY. This is a cytogenetic thing. This is a point mutation typically in P10, which is a tumor suppressor gene, which interestingly also elevates your risk of autoimmune disease when you're heterozygous PTEN. Okay, so we counted lymphocytes in all the control cases and all the autism cases, including a few genetic conditions, which I just showed you. And the vessels with the most lymphocytes were plotted, both in the controls and in the autisms. And there's about 15 case examples, as I showed you above, and we did an automated counting of the lymphocytes. And you can see that there's many autism cases that stretch up beyond the highest control here, including the 2 autism genetic forms. And this was about 2/3 of the cases of autism, 65% roughly, that had these lymphocytic cuffs in the perivascular space. Then we quantified the various subtypes of lymphocytes using a stain, which is brown here, targeting just T cells versus B cells, or cytotoxic T cells, the CD8 cells, or the CD4, which are called helper cells. But some of them do repress the immune system, and that's their role, just to limit autoimmunity. And granzyme is one of those effector molecules that degrades when it targets a cell. So we found predominantly the T cell and not the B cell. So the T cell receptor bearing cells, not the antibody producing cells. And you can see that there are many more of these T cells in the autism that control cases. We saw predominantly the cytotoxic CD8 subtype, which I showed you at the beginning, right, is the one that acts to damage something. Okay, and then we also found this unusual perivascular feature in very small little foci. So those lymphocytic cuffs are scattered. They're not uniformly throughout the brain. And these were also scattered in small, rare blood vessels. We could see these little round structures, right, of various sizes that were, and sometimes that was broken down into this debris. Interestingly, that marked with the astrocyte, which I didn't tell you what that cell was, but it's a cell that's sort of the support cell of the brain. It's involved in transporting glutamate out of the synapse so that you don't get flooded with too much glutamate. It clears out the potassium, which is involved when a cell fires active potentials. It's normally inside the cell and goes out when a cell is firing. That cell, and it also produces a barrier here between the blood vessel and the brain. And in the cerebrospinal fluid, which is the fluid that surrounds the brain, it's in the ventricles, it's on the surface of the brain, but it's also around these blood vessels. That's where we found astrocyte, GFAP positive, and I'm starting to call them blebs because essentially they look a lot like the blebs that were coming off the cancer cell. And it marked with multiple, astrocyte markers, and it was much more abundant in autism than controls. And the quantity of that astrocyte-derived debris was linearly related to how many lymphocytes you have. So if you have a lot of lymphocytes in your brain, you're going to have a lot of these blebs in your brain if it's one of these particular types of autism cases. So that really argues a little bit for a potential, we haven't proven this, but a potential cause and effect relationship between the lymphocytes and possibly the generation of that bleb-like debris that we're describing here. So I'll show you the model at the very end. So is there reason to think the GFAP coming off on these blebs that goes to the cerebrospinal fluid, works its way into the blood, as a variety of proteins coming out of the brain do, which you can track in the case of Alzheimer's and other diseases, is that elevated in autism? And interestingly, people had already looked at that. You can see high levels of GFAP in the CSF in autism. You can see it in the serum elevated. And in this case, the GFAP quantity actually correlated to the severity of symptoms. And here's another independent study showing elevated GFAP in the serum in autism. So that's a biomarker. Okay, so I'm just going to summarize the pathology here and then wrap it up. So we think that the T cells might be getting into the brain and actually eliciting an attack based on their T cell receptors of this astrocyte barrier that's created between it's kind of a blood-brain barrier, but you could also say CSF brain barrier. So lymphocytes and other things have to get through that barrier to get inside the brain. And what they're doing is they're attacking this barrier and generating, we think, this astrocyte bleb as a cytotoxic reaction. And this is somewhat analogous to the cytotoxic T cells that attack oligodendrocytes in multiple sclerosis. And here, they're not targeting the astrocytes to produce plebs. They're instead attacking the oligodendrocytes that produce the myelin that sheathe the axons in the white matter of all the neurons. So that's when a synapse transmits to the cell, they have to transmit that along these long distances along axons, and for that to work efficiently, you really need to wrap tightly these sheaths around the axon so that you don't lose that signal as it's running down the axon to the next cell. And what happens is there are some antibodies being generated in MS, and there are some T cells, and actually it's predominantly T cells if you go in there in addition to macrophages, stripping the myelin off these axons. And these are just images of some of these demyelinated plaques in the white matter and lymphocytic cuffs and the breakdown of the myelin here. So generally what I've told you about is that there's a variety of symptoms present in autism. We think that each symptom is going to map to a different brain circuitry, but we think that the same molecular pathways may be acting at each of these circuit sites to cause these various problems. Ultimately, if you wanted to target these symptoms, just for example, like risperidone targets aggression, you would want to understand what circuits are being, impaired, so that you could design medications to reverse those circuit defects. Or alternatively, you could use gene therapy approaches or the chemogenetics that I just told you about, turning up or down circuits to kind of improve some of these major symptoms. But then what I just showed you is that a large proportion of autism may involve an immunologic process that really had not been recognized previously. And the reason I think that is because people that are neuropathologists had not been looking at the brain with this sort of knowledge that there might be an inflammatory reaction. It really forced us to sort of look carefully at every little feature to make sure that that is present in a control and not, if there's something that exceeds the normal control patterns that we see. And that really is what helped us discover this pathology. And, it's really, a starting point, I would say. But I think it's a good lead now that we really want to pursue further. I think that's it for me. I'll give it back to you, Endre.
Thank you, Dr. Anderson, for such an in-depth view of your research. And I've seen other versions of these same findings, and I gotta say this is a lot more digestible. so thank you very much for helping us to understand the basis and implications of your research. I do have some questions. That have been coming in.
Okay.
And just to recap, as you were, clearly summarizing at the end of the presentation, you are describing, like, just broadly speaking, two forms of potential, autism types, so to speak. One that is something that we can call strictly genetic, let's say, and another one that is like more inflammatory, like a process that may be due to an autoimmune process. I guess one of the questions is, how could someone know whether the nature of the child's autism is either strictly genetic or, let's say, whether it is inflammatory?
So there are genetic tests available. You can do whole exome sequencing. You can do there are these arrays where you can look at copy number variations. And this is available in many of the laboratories. Our hospital provides it, as do Children's Hospital, for example, in Boston, to look at these deletions and duplications. The advantage of doing that is that you can sort of join families and get a little bit of a better sense for what symptoms might show up and what you need to watch out for, and possibly they may respond better to certain medications. It's true in the world of epilepsy, certain genetic forms respond well to one medication and not well to another. And so you can sort of save time and not have to sort of go through trial and error there. So I think that those can be assessed and identified now. It's a relatively rare subset, but I do think that there's value in identifying those. The inflammatory subtype which seems to be the more frequent subtype, which I do think will ultimately feed into those circuit defects in those pathways. Otherwise, you wouldn't have a behavioral problem. Unfortunately, I have to say that there is no routine diagnostics that's in place. And in fact, that's something we want to build and put together. And I think we have great clues, right? We think GFAP is a very good potential biomarker for sort of that process that I just showed you. There's going to be other molecules within those blebs that will also serve as biomarkers. I think that that's one thing. Eventually, as we understand what the T cells identity is based upon their T cell receptor sequences, and maybe even what they're reacting to, because we think that that's reacting to a very specific set of molecules, and it could be the same in a majority of the cases, but it could be different in different cases too. As we start to understand that better, there are assays that you could do in the peripheral blood, but again, those are not available anywhere that we know of. And certainly the markers are in the cerebral spinal fluid as well. And the cytokines and chemokines, but here again, it's not kind of part of the standard diagnostic approach to autism.
Mm-hmm. Yep. So you mentioned at the beginning of your presentation that the UBE3A gene imbalances are being inherited from the mother for the most part. And I guess the question is there any way to prevent the UBE3A imbalances from happening?
So too little produces Angelman syndrome, too much produces this autism disorder. As you understand where it's acting and what it's doing wrong, when there's too much, you could actually theoretically inhibit it, inhibit that process. And we sort of have, we already understand a lot about how that's happening. We're going deeper and deeper. And in theory, you could do a sort of a small molecule screen to inhibit that. But as of now, there is no medication that will target those pathways.
Mm-hmm, mm-hmm. Another question that I have is could seizures be, since you mentioned that seizures was kind of related to to some forms of autism, could seizures be some sort of a biomarker for determining whether the autism may be really genetic or inflammatory? I mean, what are your thoughts on that?
Good question. I'm not sure that it would necessarily tell you it's more of a genetic thing. There are genetic forms of epilepsy, as I went through, and maybe you are enriching a little bit for genetic things with epilepsy, but nobody's really proven that yet. That's just a speculation on my part. But I would not be surprised at all if the inflammatory autisms may also have epilepsy. And actually, interestingly, we have evidence of that in our paper. Now that I think about it, because one of the questions was, is the epilepsy producing the lymphocytic infiltrate? Like in reaction to the seizures, and it is not. So if you plot the data for lymphocytes versus with seizures or without seizures, and we had controls with seizures as well, other forms of epilepsy that had passed away due to SUDEP. There's no correlation of the lymphocyte numbers to the presence or absence of epilepsy. But that does mean that there are some cases of lymphocytic infiltrates that also have epilepsy. So I don't think you can segregate them this way.
Yep. I guess that before we move on to the questions about the potential inflammatory processes in this potential autoimmune process, I think a question people have is regarding the if it doesn't have a strictly genetic base, could we also understand the inflammatory process as an epigenetic phenomenon, where you're somehow by virtue of the environment and other causes be influencing your genes and not necessarily having these forms and then that triggering some form of inflammation?
I certainly think that the inflammation itself could be perturbing the epigenetics. That's sort of my favored view. Also, you have to ask yourself when. When is this important? Is it important very early on, when the brain's epigenetics is remodeling, right? So when you form a brain with these cells, they haven't yet even formed synapses or dendrites. Those cells are maturing, the whole circuitry is maturing, all these molecules involved in how a neuron works are becoming expressed. That's a huge shift in gene expression that's happening, and that could theoretically be perturbed if there was something happening in utero or even early postnatal. The other question, and is any of that as an epigenetic perturbation, which I do believe in, and there's pretty good evidence of epigenetic perturbations in the methylome data in the postmortem brains. So methylation of the DNA itself, which is a really nice kind of permanent reflection of gene expression features that don't fluctuate so quickly as transcripts do. That there's pretty good evidence that that is perturbed, and in a large proportion of cases, and nobody's really correlated the inflammatory state to those epigenetic changes, but I'm sure that's coming very soon. But I think that that could very well be true. And certainly, yes, epilepsy itself, as we just heard, can change expression of genes like cerebellin-1, which can break synapses and lead to behavioral impairments. And that would be sort of a ongoing kind of acute effect of seizures. Now, it's also possible that there is some component to the inflammation ongoing, as we've just seen. The cytokines are being released, chemokines are being released, floating through the CSF to all the neurons, and then that is perturbing their gene expression to cause dysfunction as well. So I think that there's, those are all possible, and those are all kind of in the models of how we're thinking.
So is there, you were kind of covering another question that I had, but I would like to tie your answer with yet another question, which is, it sounds like, the most likely, possible cause or stage rather of the development of a person could happen when the brain is formed, which is during the gestational stages. But then at the same time, there's other like well-known conditions like autoimmune encephalitis, for example.
Yes.
That may happen in adulthood, right? So to what extent is this process similar to autoimmune encephalitis, or is it the same thing and it's just simply expressed at a different stage in the person's life?
Autoimmune encephalitis, there's various forms of that, and they target different molecules. And they certainly are very impaired, right? These individuals are severely impaired. They can have psychiatric-like symptoms. They can have kind of dementia-like symptoms. It could be, and we don't know, what is different? Well, one thing that's different between the autism cases is it's not autoimmune encephalitis-like. It's not attacking the neurons. And I've seen cases of that post-mortem, so it's very different. It's not MS-like, multiple sclerosis-like. It's not Rasmussen's encephalitis-like, which is an epilepsy where T cells are attacking cortical neurons. It's not like that. So it's a different target in this case. So it could be that the concentration of inflammatory mediators is happening in a different compartment. That could be one of the reasons for the difference. I do think that analogies to therapy that are not super targeted, but sort of more general, could be made with these other disorders. Like, how do you treat multiple sclerosis? How do you treat Rasmussen's encephalitis? You may want to use the same kind of therapeutics that are used in those, because none of them are highly targeted. They're kind of generally repressing the immune system in the brain, but also other organs often.
Yeah. So tying up the answer with another couple of questions I have is, so could history of other conditions like the ones that you have mentioned, like MS, multiple sclerosis, or maybe other kinds of autoimmune processes, could that be somehow related to the probability of developing autism. So I'm guessing, from your answer, they may not be necessarily related, since it's kind of a different, target. But what are your thoughts on that?
So one thing that we do know, and we've actually had a case of this, we do know that inflammatory bowel disease pathology, true inflammatory bowel disease, where there are T cells invading the gut epithelium, the gut lining, that can coexist with autism. And sometimes it's just the symptoms, GI symptoms, not full blown, something diagnosed as inflammatory bowel disease. So that coexists. And, how do you think about that? Well, that's an inflammatory T cell based adaptive immune system based process happening in the gut. And we are working on evidence that it's also coexisting in the brain, the pathology we just described. So if that were true, you might imagine that it could be a complex autoimmune reaction to many different epitopes, but it could also be a shared epitope, right? It could be that something expressed in the brain is also expressed in the gut. And in the case of cancer, where you get a cancer and the immune system is attacking the cancer, and then you suddenly get an autoimmune disorder, there's a variety of them. People think that it might be sort of a shared epitope where that protein that it's starting to react to is in that other organ system. So I think that some of them are because of shared epitopes. It is true that in autoimmune disorders like type 1 diabetes, you get a sort of a building cascade of additional epitopes over time. These are the protein antigens that are different proteins that are sort of reacting to. And what happens is, as you damage the cells with the first thing that it's targeting, those damaged cells release these blebs and other bits of protein, and that actually goes into the lymphatic system, into the lymph nodes or the spleen or whatever, and that can then elicit a reaction to new proteins that are being presented. So you sort of get a building up of other autoimmunities, and then some of those may cascade out into organ systems that also express those newer cascaded antigens that it's reacting to, and then you get suddenly an autoimmune disorder to other things. And I suspect that autism will be like that, that they'll be cascading out of different autoimmune things because of the damage that's happening.
Yeah, I just want to clarify something, because I think it's important. So I'm actually interested in the way that you kind of quickly jumped to IBD, right? To Crohn's disease and also colitis, because I think a potential question is whether these autoimmune processes that you're observing resemble any other already known autoimmune conditions, such as there are several. I mean, certainly UC and Crohn's disease, but also like rheumatoid arthritis or psoriasis, psoriatic arthritis. So the fact that you're specifically pointing at the bowel, for the potential origin, would that imply that there's something that is specific about the mechanism in IBD, Crohn's disease, also colitis, that is not probably resembling to the process, to other autoimmune conditions?
I mean,
maybe?
The question is what comes first. I'm not sure. It could be that people, autism comes quite early, right? Very, very, very early. So I'm not sure that I would necessarily point to the inflammatory bowel disease being the driving force. It could be. I'm not saying that it's not, but certainly I just really don't know, honestly. I think that they're coexisting. And yes, I think that the immune reactivity could be happening in both sites, and it may be the same kind of population of autoimmune T cells, or for example, or B cells even. The other thing that's quite interesting is that, at least in multiple sclerosis, where there's mostly cytotoxic T cells in the brain, they find that the B cells are playing a role, because if you limit the B cell reactivity with a CD20 targeted treatment, you actually make multiple sclerosis better, actually. So it could be that these pathways are sort of interacting with each other at some level, possibly in the lymph nodes or something. But yeah, the gut, it's hard to say what is the driver, but it's certainly, we need, I'd like to keep an open mind, for sure.
Sure. So if you could, at this point with the knowledge we have, we could narrow down into the hypothesis regarding the T cell activation. What would you say that is the most likely cause from your perspective?
I think it's going to be some specific antigens that the T cells are reacting to. It could be and that it's autoimmune. That's my favorite idea. It could be still a virus that has not yet been discovered. In the paper we published evidence that we stained for a variety of known viruses that are reported, and we didn't see any of the sort of common CNS viruses in any of the samples. So that still remains a possibility. But I sort of like the autoimmune idea.
Yeah, I have a question here from John Rodakis, which I think is interesting. He's asking, what is your theory on why T cells may be attacking specifically astrocytes? If they could just go and probably attack other cells. I mean, why specifically astrocytes?
I think that there's some kind of epitope uniquely expressed by astrocytes that is what they target.
Okay. Another question from, let's see. So I think we have covered most of these. So
okay.
I think we have done a pretty good job now at answering most of the questions. So this has been really, really interesting and great. So I think I just have this last one. So from a couple of parents, do you think that any of these potential pathologies or autoimmune processes could be modified by things like diets or things like supplements or even biomedical treatments? And I think that this is kind of tied up with these potential treatments that are being studied currently that have shown like very important and encouraging results, like the one from Dr. Jane Adams and Dr. Jürgen Hahn, who we recently had the opportunity to interview, that are looking into like microbiota transfer therapy, and that also have demonstrated how diets or certain supplements, vitamins, etc., they can really alleviate some of the autism symptoms. So what are your thoughts on that? I mean, what things beyond actual treatments for these could help to alleviate some of the autism symptoms and probably alleviating the autoimmune process?
I mean, I think that one can look at sort of autoimmune disorders in a very general way and ask what has been effective in those. And it's true that some diet modifications improve symptoms in things like ulcerative colitis and IBD. Gluten enteropathy is an issue that could simply be coexisting, but if it gets the immune system activated, it could worsen the symptoms theoretically. What is it? Vitamin E is now prescribed by, it's D, sorry. Vitamin D is now prescribed to multiple sclerosis patients. And there's a correlation in how much sunlight you get. So the northern hemisphere seems to have more multiple sclerosis than the southern hemisphere. It's sort of that being near the equatorial line. So essentially large amounts of sunlight may be effective in enhancing your immune system. So I think that I would recommend people look at what people do for other autoimmune disorders. And at least in treating the CNS for an autoimmune disorder, I am beginning to, sort of collaborate with colleagues that run the multiple sclerosis clinic to get their viewpoint, because I think that some of the medications used in that clinic will eventually, the problem is many of them have very severe side effects. And you really, really have to be careful, and you have to be followed carefully because you can develop other diseases, by virus infection and things like that, some of these therapies. So, I think it's going to be, and, certainly the other thing is that, this stuff will over time be reviewed by a IRB, and, there'll be clinical trials that really demonstrate efficacy, because they're expensive, they're dangerous, some of the medications. But the key is that there is more evidence building for this kind of mechanism, and then people will come up with therapeutic solutions over time. I'm confident of that. One other thing I want to just sort of encourage everyone to consider, and ideally sharing with everyone that also might be affected, that may not have heard this talk, is that it's a very hard thing to think about at the time, but if you hear of a family where somebody has passed away, to do this act of donation, because it makes such a difference. And I think it's really going to be transformative for the field. It's transformed many other fields in brain disease by the ability to look at what's actually happening in the tissue.
Yeah, definitely. We will definitely promote the contact information of whoever would be willing to make these donations for a deceased relative. I mean, this is critically important. I mean, and everything that you're doing, Doctor, is like absolutely groundbreaking and essential to really understand the mechanics of this. I just have one last question for you. I know that we're past the hour, so I really appreciate your time. A couple things. So one is, like, there are obviously some treatments. We know that microbiota transfer therapy has been studied for IBD, for example, and that's currently in research. We know also that IVIG, that is also used for things like autoimmune encephalitis and other things. And also we know the use of biologics that are very, very commonly used for autoimmune diseases, Crohn's, also colitis, RA, etc. So what are your thoughts on these types of treatments to approach this autoimmune process?
I really believe that I think there needs to be a step-by-step process where we get biomarkers of this pathology, right? We need the biomarkers of the pathology. Once we get the biomarkers of the pathology in hand, I think that we need to really think carefully about which of those medical approaches have been effective in autoimmune disorders that are behind the blood-brain barrier, that are sort of, well, actually, this is right at the blood-brain barrier, but in the CNS. And we do need to start these clinical trials. But right now we don't have any marker to show that it was effective yet. So I think that's a key step, is to get the biomarkers that we can say, yes, look, this medication changed this biomarker, because the behaviors are measurable, but some of them may not be rapidly reversible, right? If you're sort of had difficulty learning, you may have to add supplementary learning once you clear out the inflammation, if that's sort of causing ongoing problems. It could be that an EEG signal would be good, if there is some interictal spiking or even truly epilepsy driven by this. That certainly would be a relatively readily measurable biomarker. But I think we need a tissue, not a tissue, but a sort of a blood and maybe CSF at the beginnings, but ideally blood biomarker of this process. And then we should start to use those, I think.
Yeah, could be something that, like GFAP, the protein that you were just referring to. I mean, that could be an option. Dr. Anderson, I mean, your research is obviously giving a lot of hope to families, and it really highlights the fact that autism is medical. I mean, there is an underlying, thing that may be going on that we're still investigating, and it could be certainly in many ways. I mean, clearly an autoimmune disorder that could be potentially treatable. So this is very, very encouraging news to families. I just want to thank you so much on behalf of all the families that are watching, for all your work again, and I really hope that you give us another interview when we have more information on your research and on all the things that you're working. So thank you so much for your time.
Thank you. I enjoyed it too.
Thank you so
Thanks
much.
a lot
Thank
for
you.
all you do.
Thank you.